apparently some people eat like just a slice of cheese. I do that.
Oh yeah. Or like whatever. Okay, well, maybe sometimes like the cubes, but I don't know. I gotta be able to make that. I feel like that's just cheese. No, that's really nice design.
I thought we were gonna split off too. Like that's just like fucking like the number of split.
Ratings growing everything still said he felt sick. They decided to not tell anyone until.
How do you feel sick after just being a black lady?
Yeah. Mine was talking about him too.
He was there first, assistant coach with Asher, and then he was my freshman fall assistant coach. I've had 3 assistant coaches here, and I've been here 3 semesters. Well, this is my 4th. Yeah.
So you really got to see it.
And then
and
I don't know how far it was exactly. That's a trigger word for us right now.
What, graphic? Yeah, don't talk about it. It's a trigger word. Yeah.
That's a trigger too.
Yeah, that was awful too.
Like, we literally got like 8 through 8 or something like 8 to 3. And I was on the 5th run.
One person has to do 2 quiz of the 20
Put it back in. So you can wait there. And I was thinking that she was like, I swear, when I was older, like people were like a whole set in like, and I'm like, how? Like now I'm just like, I don't have to change it. And me and Sophie didn't touch a red one. Like we would do the exact choice. And we would still a full step behind it. And I'm like, how did I even?
People just don't do it.
It's just the thing it pops up on first. It's a camera. Alright, you guys know. I'm just gonna keep. Okay. No, I'm just like, no, I'm distracted.
Really?
Oh, okay. At least I can do it. I'll be distracted. You've never had me.
So funny.
I was close to 78.
Oh, yes.
Yeah, like 94. Why do that?
It was actually supposed to be written. So like in the moment, I was I think I got two.
Oh, I see.
I don't know, like there's something about this one first. And I there, but like just in my head, I was like, oh, it's good. Like she like showed it to me. Her new one. How old are you?
And she told me that if I did my service,
I'm just waiting to get money again. On like a certain day, it was like a certain size.
if you go on Friday the 13th,
I don't wanna go to.
okay, well, we think if you on Friday the 13th, and I'm like, oh,
Does anyone work in IT or anything like that? Because it's like frozen and I can't unfreeze it. So sometimes there's someone who actually knows how to unfreeze these things. Anyone? No? Okay. I guess the other thing is, I know there's a number to contact IT, but I can't locate it. Does anyone have that handy by any chance? Okay, how about this?
Yeah, for you.
Can someone look it up for me? And then we'll try and get some IT support here to kind of unfreeze our screen for us.
Is it 517-417 help? 4357, that's what I'm looking at.
Yeah, I'm not sure what the number is.
Want to call
Sure. We're in Peelle207.
Tell us. Like Jen is like our whole friendship mistake. Like Jen is like going through. Yeah.
Yeah, no answer.
Okay, well.
I'll look for another number.
Well, thanks for trying.
Sure.
Because like normally I would turn this thing off, but
Yeah, can you just unplug it?
restart, but like I don't have that. All right, let me try it. Let me try and power off. Well, actually, I don't know if I can power off from over there.
Yeah, that didn't work either.
All right, we're gonna use your, I don't know if you all have a laptop so that you can kind of follow along with the slides that are on Blackboard. If you don't mind, opening up those slides and I will walk you through those. So it looks like all of you have a laptop except me, so I'm going to use your laptop. So how's that sound? All right. So again, if you haven't looked at the slides before, they're on Blackboard. I wish I could show you where they are, but I can't remember what the menu is called here on the left. It's like instructional stuff. But can you locate the lecture slides? Yes? Okay, good. So we're on chapter number 4. Can you open that one up? Okay, awesome. So, I'll stand here and kind of go through the first couple that I can see, but then after that I'm going to take a look at what you have there to kind of keep me on track here. But chapter number 4 is about microbial growth. And so the idea here is that by understanding the conditions that are required to grow these microorganisms, we'll get a better understanding of how we can control their growth. Remember, from a health perspective, it's more important to be able to prevent growth than it is to kill microorganisms. Okay, so that's the emphasis for that kind of explains the importance of chapter number 4 here. And then for the second slide, you're going to see information about Robert Koch. Koch was a German physician who actually ended up being awarded the Nobel Prize because of his work in understanding the importance of these microorganisms as causal agents of disease. And later on in our class and in our textbook, we're going to learn about Koch's postulates, where he actually came up with a list of steps in order to establish, that one organism is the direct cause of a particular disease. Now, there's different ways that you can grow these microorganisms. You can grow them in liquid, and we kind of learned about that when we learned about spontaneous generation. You could take water and add like hay to it to make an infusion, or you can grow microorganisms on a solid medium, like what we're doing in the lab using petri dishes. There's some advantages to growing them in a solid surface versus a liquid state. A lot of microorganisms require oxygen just like you do. And so if you put them in water, there's just not enough oxygen in there. There's tricks around that, like in our laboratory. We have this device that's called a shaker that will actually mix up the water, the liquid, in order to get more oxygen into the liquid. But back in the old days, when Koch was doing these experiments, they didn't have these kind of shaking devices. So instead, they wanted to try and grow these microorganisms on the surface of media where the microbes would be continually exposed to oxygen. And then on the bottom of the slide, you're going to see the name of a woman. What name is that?
Pretty Hess.
Yes, okay. She's the one that pointed out to Koch that, hey, why not use agar as the solidifying agent? So agar is basically a gelatin that's derived from algae. And you can use that as a, as a solid medium on top of which you can grow microorganisms. And then you can actually add nutrients to that agar as a source of food. Okay. So once that system was established, then the idea is that people could grow these microorganisms. Jeez, this is horrible. So there's I did highlight and read a word. It's cultivating. So I know that may seem a little odd at first. Normally when we think about cultivation, we think about growing plants on a farm. But a similar kind of concept can be used with microorganisms. Our farm is the petri dish on top of which we can cultivate, we can grow these microorganisms. So cultivating, culturing is another term that you're going to hear quite a bit. So cultivating, culturing refers to growing. Next slide, I guess number 3 there, shows us that process where bacteria can multiply. So it's like mitosis. It's called binary fission. So one cell will basically split to form 2 to 4 and so on. And the numbers will increase exponentially. Yeah. And then let's see what else we got there. Oh, we got the generation time. So this is the amount of time. It could be in like minutes or hours that are required in order to double the number of microorganisms, double the number of cells that are present. Okay, so that's our generation time. Sorry, got something in my throat. All right, what's the last highlighted line there? Right there. I can't read that one.
Microbial growth is defined as an increase in the number of cells.
Oh, that's a good point there. Because normally when we think about growth, we're thinking about increase in size, right? So like when you were a baby, you got bigger, and look at how big you are today, right? Microbes, we're talking about not increases in size, we're concerned more about numbers. So growth, when we talk about bacteria, we're talking about numbers, increases in numbers of cells. Oh my gosh, I'm dying here. All right, next slide here. This one shows us growth in the natural world. So like what we see outside of the laboratory. And we see microbes growing in these films of polysaccharides referred to as a biofilm. And this, we mentioned or learned before, can happen, it could be happening in your mouth. That's what we result in dental plaque. These films are seen outdoors in our streams and rivers that kind of creates this sliminess that we find on rocks and pebbles that we pull out of lakes and streams. So we also see biofilms in your bathrooms, like in the sinks, the shower walls, that they're a little slippery, inside the toilet, where you're not touching it, you're not supposed to touch that. But wherever there are microbes, they'll create a layer that they will adhere to, then that's our biofilm. All right, next slide should be about growing microorganisms in the laboratory. All right, let me see what we got going on here. Put your hand down. Next slide for me. So the next slide, we're going to learn about how to grow what we call pure cultures in the laboratory. So what we mean by pure is when you only have a single species. Pure is single species. So you can imagine when you start isolating microbes last week from your body and also from the environment that surrounds you, that you were isolating, you were swabbing surfaces and you were picking up hundreds of different kinds of bacteria, and you're plating them, you're putting them into your petri dish. It's kind of hard to study, like 100 different kinds of microbes, and it's a lot easier to just study one at a time. So this week in lab, we're going to be trying to, isolate individuals in order to grow and start a pure culture of just a single microorganism. Okay, then we can do further studies with that. And in order to do this process, we're going to use what's referred to as the aseptic technique. All right, any thoughts about what we mean by aseptic? Anyone? Sounds kind of like a septic tank. No, it's the opposite of that. Sorry, got something in my throat. Help me out. Aseptic, go ahead.
I don't know, maybe we are now going to be just working inside of those, I don't know, things that we have in the labs. Probably maybe to just limit the amount of bacteria that can just pour onto our petri dish from the air, and we are just working maybe at the bad thing.
Yes, that's very good, Blessing. So we're talking about trying to create like sterile environments.
Yeah.
So aseptic would be like excluding other microorganisms that may be present, like as Blessing described, that are in the air or on the table or even on your hands. So in order to generate these or create these pure cultures, we want to exclude other microorganisms from entering into your petri dish. So we're going to be using aseptic techniques for that. All right, Anna, what's the next slide? Oh, okay, good. Yeah, next slide shows us a fun picture of a petri dish. And you're going to see all those little round colored, kind of slimy looking blobs of bacteria. And each one of those is referred to as a colony. And each colony is actually derived from a single progenitor cell. So one cell multiplied by binary fission many times. So from one cell, we ended up with like 1 million cells, and they gradually spread out from that original cell to form a circular colony. So all of those are derived from the same cell. So because binary fission is asexual reproduction, that means all those cells that are in that colony are genetically identical to each other. Well, one nice thing about that picture is that you can see that there's different kinds of bacteria present, right? Because you got different shapes, different colors. And one thing that we're going to be doing on Tuesday in the lab is looking at your petri dishes, that some of them may look kind of like that. And we're going to look at the edge for the margins of those colonies, which can kind of give us information about, you know, how similar or how different those different species are. All right, animals, next slide. Okay, here we have more information about agar. So remember that, you know, they can be isolated from algae. And you could actually purchase this on Amazon if you want to. You can actually go on Amazon and purchase yourself a bottle of dried algae, or agar, dried agar. It comes as a powder, like a white, clear, you know, white powder. And then the idea is that in order to, you know, create what you have in your petri dishes, you want to, you know, weigh out a certain quantity, add it to water, and then heat it up in order to sterilize it. And then the idea is when you heat it up, the agar, which is originally a solid, will turn into a liquid. And it's that liquid that you pour into those petri dishes, and then as the temperature cools, it will solidify. How many of you have made Jell-O before? Yeah, same process. You start with the powder, you add the water to it, you heat it up till it dissolves, you pour it into your mold, and then watch it solidify. same process. All right, Anna, what's next? All right, next we're going to learn about what we call the streak plate method. Streak plate method. Okay, here we go. Let me use the board here. I'm going to draw a circle here on the board, and that circle is going to represent a petri dish. We're gonna actually do this one in the lab this week here. Streak plate method. Just for fun, some people refer to this process as streaking. There's always fun connotations of these kind of things, right? Okay, and then in this process, the idea is that we're going to want to try and take you know, a lot of bacteria and spread them out over the surface of our petri dish in order to separate individual cells from each other that can give rise to colonies. Okay, so in the lab tomorrow, everyone's going to be working with what we call a metal loop. So it's basically a piece of wire, metal wire, and at the end of it, you know, it's been twisted into a circle there, and then at the bottom you have a handle that, you know, you'll grip with your hand, and that little loop there at the end is what you're going to be using to pick up and move bacteria across this petri dish over here. Now we want to use aseptic technique. So again, what does that mean?
Sterile.
Yeah, like sterile conditions, right? And so we want to actually sterilize our metal wire before we pick up bacteria out of your petri dishes. So we're going to do that using our Bunsen burner. You like that cool looking flame there? Hey, I want to see you do better. All right, so we're playing with fire on Tuesday. So in the lab we have some hair ties, but just kind of be aware of what you wear, so you don't catch on fire. Hey, we got our paramedic here, right? Just in case. Good. All right. So when you put metal into a flame, or expose it to flame, the tip of the flame is the hottest point. So that's where you want to put it. Put the wire, not at the bottom. At the very tip is the hottest point. And what do you think is going to happen to our metal wire once you put it at the tip of that flame?
Turns red what?
What?
It turns red.
Yes, that's right. So there may be some residue left over from wherever you used it before, and it will ignite. So you might see a little flame there, but then the wire itself would turn bright orange red. Don't touch it. It's hot. Okay. All right, and then the idea is that once you have to let it cool down after you sterilize this wire here, and then the idea is that you're going to pick up some of the bacteria from your petri dish that you used last week, and you're going to transfer it onto a new fresh petri dish over here on the left here. So when you look at the slide there, one more slide. Yeah, there's a couple of different patterns you can actually use here. We can try them out. So I'm going to show you a different kind of pattern than what you see in your textbook there. So try and imagine my marker here is the loop. Okay, try and imagine my marker is the loop. You open up the petri dish from last week. There's some interesting bacteria there. You're going to pick it up in that sterile loop there, and then you're going to put it on our fresh dish, and in a continuous motion, you'll start at the edge and then drag that loop across the surface and like little squiggly lines there. So that the idea is that when you where you place the loop on initially at the surface, try to imagine there's a million bacteria there, but as you drag your loop across the surface, there's fewer and fewer bacteria left behind. Okay, so you may have 1 million there, and then maybe 100,000 over here. Okay, what if we just stop there and let this grow? What are we going to see? Not colonies. We're going to see if we have 100,000 there. Well, okay, let's say each one of those forms a colony, but they're all packed together. What's that going to look like? It's going to look like a smeary mess. it's getting all smeary. So we're not getting distinguished colonies. So the idea is that we need to do this process again. So you're going to take your wire loop and re-sterilize it. So we get the 100,000 off of this loop here, and then we're going to come back over to our dish. And then we're going to start over on this side, and we're going to go zigzagging back and forth, dragging bacteria back towards the center. So this time we're just taking a small amount, maybe 10,000 over there, spreading that 10,000 out across that wedge there. And then repeat the process. Sterilize, let it cool down. Take your loop, drag it across part of that, that second wedge, make a new wedge, and then repeat, repeat for the fourth time. So, are you familiar with the concept of serial dilutions? Yeah, that's the concept here. we're slowly diluting the amount of bacteria across the surface of that petri dish. So tomorrow you're going to do that 2 times. You got 2 tries. All right. Any questions about this streak plate method? Streaking. All right, so let's see what's on the next slide here. Help me out, Mallory. What's on the next slide?
Mine won't load.
Oh my
That's why
God!
when you're like, do you have it?
You
I'll
can't
download it.
download.
I download it and I click it, and then all these you have to do one laptop.
Like it's a PowerPoint file, right? So you need PowerPoint. I think that's why you can't open it. But doesn't the school provide that? Or?
This is a newer laptop, so a lot of my old stuff is not on it yet. So.
Let's look into that later. All right, what do we got here? Okay, so try to imagine we do the streak plate method. We get colonies at the end. You know, each colony is that's a pure culture right there. Okay. Then we can store, because remember we can't be in the lab every day. So you've got other more important things to do, right? So we can temporarily store those petri dishes in our refrigerator. So that's good. But eventually, we just can't leave it in there for the whole semester. Eventually, those bacteria will die. So if we want to preserve those cells for a longer period of time, then we can actually put them in these low temperature freezers. So the slide says minus 70 degrees. That's Celsius. Anyone know what degree Celsius does water freeze at? That's 0. Okay, so we're talking about minus 70. Okay, so that's really cold, right? Okay, so that's the idea here. All right, next slide. Okay, next one shows us a growth curve. All right, let's get that one on the board here. All right, let's start off with labeling the 2 axes of our graph. So what do we see over here on the left?
Number of cells.
Yep, very good. And then on the bottom, time, very good. So we're going to start off, at the beginning, which is on the very left, with very few bacteria, right? Okay. So the numbers are initially low. Okay, and then you're going to see that we get the graphs sort of like this, and we can kind of divide them up into different stages. So let's take a look at this first stage here. Okay, what do you see it labeled as? Lag phase, yep. Okay, so this was actually even before computers. even back then, they had this term lag. so what do you think that means? Yeah, there's a delay. That's right. So even though bacteria maybe provided fresh food, it takes them a while to start multiplying. So that's our lag phase. But then look at what happens after that. Okay, and then we have what phase after that?
Log phase.
Okay, then we got our log phase. So now we have like an explosion of rapid growth. Our numbers increase rapidly in a relatively short amount of time. Lots of binary fission taking place. Now, even though they're multiplying, our bacteria don't live forever. So some are also dying, but the numbers that are created greatly exceed the numbers that are dying. But things don't last forever. And then we reach this phase up on top, and what's that one labeled as? Stationary phase, very good. Try to imagine, this is like in a petri dish or maybe in a flask where you're growing things in a liquid. The food is starting to run out. Okay. And then we start seeing that the number of cells that are dying are about the same as those that are being created by binary fission. So as a result, the number of live bacteria reaches a plateau. And then we have our next phase. And what do you see that one labeled as? Death? Okay, that's pretty much, that's kind of self-explanatory there, right? The food is pretty much running out at this point. And then?
Prolonged decline.
Prolonged decline, okay. Unfortunately, death does not always come immediately, right? It can be dragged out. Oh my gosh, I hope I have a quick death. All right, let's see how we're doing on the slides here. What's next? Okay, now we're looking at some of the environmental factors. So, similar to the way that there are different situations that can either promote your development or stymie it. Right? There are conditions under which bacteria either will thrive or they'll be prevented or inhibited from growth. So let's take a look at some of those major factors. So they include temperature, okay, so if there is bacteria do have a temperature preference. A lot of that has to do with the preference of their enzymes that are inside of the cells. And that's also true for you as well. So, one of the reasons why, you don't like this Arctic temperature outside is because your enzymes don't like it. There's actually a reason, a biological reason for these things. All right, so you may remember last week in the lab, we actually put your bacteria under 2 different temperatures. So you set 2 petri dishes. One of those dishes went into what incubator? Remember the temperature? That's right, 36, 37 Celsius, because that would match the temperature of your body, which is where you isolate these microbes from. And the other one went into a room temperature incubator because you were isolating substances from your, the surroundings that were at room temperature. So we're trying to grow bacteria that will match their preference, or hopefully will match their preference. Okay, then we have the atmosphere. The atmosphere is also a factor. So what do you think they mean by that? The atmosphere? Isn't the atmosphere the same regardless of where you go? What is it in the atmosphere that they're referring to? O2, that's right, oxygen. I don't know if you've ever noticed going up in, have any of you gone to, really high mountains? In any of the Rockies, for example, where the air is thin? It's like less oxygen up there, and you can feel it. And, likewise, have any of you ever almost drowned in water? I did. I almost drowned. There wasn't enough oxygen there for me. So the world's all the same, but there are differences in oxygen levels, right? So microbes have a preference for that as well. And then, what else we got? What are the other factors? pH?
Kitchen?
Okay, water availability, good. All right, who remembers the pH, the numbers in the pH scale? What do they range from 0 to 100? 0 to 14, okay. And what numbers are referred to as being acidic? 0 to 7, and then above 7. Basic, also known as alkaline. Yeah, basic and alkaline are the same.
Ethan has
So in your textbook, they seem to like to use the word alkaline, so you're going to see that. So just try to remember those alkaline batteries, for example. That'll help you remember that refers to basic. And then we have the water availability. Yeah, that's actually a good one. All these microbes they need water. And knowing that people have actually devised methods of controlling microbes by removing water. So like dehydration, using high concentrations of salts, high concentrations of sugar. We should see some slides about that at some point in time. All right, one last slide. Are we looking at temperature now?
Yeah, we got it.
Okay, I don't think we can do that in 5 minutes, so, 'cause there's a lot to, so next time we're gonna learn about, how we can classify microbes based on their temperature preferences. So there's like these different categories there. But, this is probably a good place to stop. So tomorrow we're gonna have more fun with bacteria. We're gonna do some we're gonna use the street plate method, aseptic technique. We're going to take a look at your dishes from last time, look at the colonies and characterize those. And yeah, there we go. All right, any questions? All right, hopefully we'll get the computer system running next time. So have a great day, everyone.
You too.